thank you very much, Yvonne. First, I would like to thank you and the organizers for the kind invitation. Here's some background information on why and what we are doing. So actually, my lab is very much interested in all different means of licensing patients' immune system to defend cancer, and this includes, of course, immune cells, CD8, CD4 cells, but also inhibitory and stimulatory molecules. And that CD4 cells play a very important role in the immune defense of cancer, has been published in a number of recent papers. I put here two on this slide, and actually one is, in fact, breast cancer, where this group has shown that the CD4 T cells in the tumor predict breast cancer survival. So this talk will focus on antigen-specific CD4 T cells in breast cancer, and the antigen I'm working on here is New York BR1. This is a differentiation antigen. It's expressed in breast cancer, and there is a study showing that at least on mRNA level, about 80% of the breast cancer specimen are positive for New York BR1. And it can be found also in normal epithelials, individual epithelial cells of the breast, but only on a low level. And it is overexpressed in ductal mammary carcinoma, and actually different studies have shown that around 60% of the cases are positive for New York BR1. So this slide should remind us on the different restrictions an antigen can have. There is, of course, the class 1 restriction, which is important for T cell recognition and CD8 T cell recognition. And CD8 cells, of course, are the ones which directly target the tumor cells and can eventually eliminate them. And for New York BR1, there are already two publications who have unraveled or identified some CD8 or class 1 dependent epitopes. And class 2 is the restriction which is important for CD4 T cells. In some cases, you can have a direct targeting of tumor cells if you have class 2 positive tumor cells, which is, for example, very common in melanoma, but it is very rare in breast cancer. Normally, breast cancer cells are negative for class 2. But there are a number of other different and very important effects of class 2 presentation. This is, on one hand, the licensing of DCs to effectively prime CTLs. There's a direct stimulation of CTLs, and there's also a new area coming up with tumor-associated macrophages, for example, which are part of the tumor stroma, and targeting of these cells may also be beneficial for an immune response. So we were aiming to identify epitopes which are presented by human class 2 molecules, and we made use of a transgenic mouse model. So we have two mouse strains which are devoid of any mouse class 2 molecules, so they are knockout for class 2, and they are transgenic for either DR3, human HLA DR3, or DR4. And using these mice, we identified epitopes from this antigen by the following way. We vaccinated the mice with a DNA vector, so this is a plasmid which codes for New York BR1, or the control mice were just vaccinated with the empty vector. So the mice have seen the global antigen, and then after 14 days we harvest the spleens, and then we go through, we use a huge peptide library containing of 20 MERs. These are 174 overlapping peptides. There's a 12 amino acid overlap, and we test these peptides for recognition by spleen cells from these immunized mice. And we ended up with actually 3 20 MERs on DR3 and 3 20 MERs on DR4. So we had 6 putative epitopes, and the underlying epitopes were predicted using the different programs which are available. So normally, class 2 epitopes are around 15 amino acids, and there was a prediction in each of them for a 15 amino acid epitope. These have been then tested. So these are again the mice, which are another set of mice which were vaccinated again with DNA. And then we used these 15 mares and tested whether the mice had specific spleen cells recognizing these epitopes, and in fact all 6 were recognized. So by this we could detect new epitopes, and by the way we also show that at least in the mice these epitopes were processed and presented. But of course we wanted to be sure that they are also presented on a human cell in a HLA restricted way. So what we used here is our T2 cells. These are human class 2 negative tumor cell line. It's a negative tumor cell line, which then has been transfected for either DR3 or DR4. And these cells were then loaded with the different peptides, and we use again the spleen cells from transgenic mice. So now we have a human cell with the human HLA molecule loaded with peptide, but a mouse T cell line which was generated in the transgenic mice. And as you can see, the respective epitopes have been recognized, which shows that in fact we have an HLA restriction on a human cell. This does not yet show whether these epitopes are really processed in a human cell, because that we have only shown in the mouse. Here we have to we did an experiment which is a little bit indirect, and I can explain to you why. We used an adenovirus vector which codes for NURBR1 and a control adenovirus. And actually only melanoma cells have the correct receptors that they can be infected by the adenovirus. And unfortunately, the available human breast cancer cells are all negative for NURBR1. That's the reason why we had to go indirect. So we transfected melanoma cells with the correct DR3 or DR4 epitope, HLA restriction. We transfected these cells and then used again the mouse cell lines, loaded these transfected melanoma cells on dendritic cells with a matched HLA and tested whether the mouse cell lines recognize these. And as you can see here, this was also successful. This is the correct, this is the adenovirus transfected cell, and this is the empty vector, and this is a control peptide, the peptide is a control, which is of course always working. And you can see in all cases, the adenovirus NURBR1 transfected cells as a lysate were recognized by the T cells. So this shows us that there is natural processing in human cells, either in the melanoma cell or in the dendritic cell. We don't mind, at least one of the human cells had to process these antigens and present it. Of course, now we would like to know whether also patients have T cells which can recognize these epitopes. And for this reason, we had a collaboration with a pathology. So there was a large number of breast cancer patients where the tumor was tested for NURBR1 expression. And those which were positive, we were able to get PBMC of some of these patients. Most patients are in many trials, so it's not so easy to get blood from them. And we had about 20 plus patients blood, and we tested, we identified the HLA type, the class 2 HLA type, and found 3 patients which were DR3 and 4 patients which were DR4. So these could then be used to test whether these patients have CD4 cells with the respective specificity. And as I mentioned, the patients all were positive for NURBR1. So these are some stainings of these patients, either of the biopsy or here of a resected tumor, which was resected later. Here are the results with the patient's PBMCs. So what we did here is we used the PBMC and cultured them for 3 weeks and re-stimulated them with the respective peptides. And after 3 weeks, we tested them either with medium control or with a specific peptide. And as you can see, all of the peptides have been recognized but not equally well. For example, here this peptide 88 was recognized by patient BC number 3, not by the other 2. This peptide here was recognized by 2 patients of 3, and so on. And this peptide here was not, there was no difference between the medium control and the peptide, but one might argue that already the recognition here with medium is a little bit overstimulated during the 3-week period. So the reviewer asked us for more controls, so we were able to get from breast cancer patient number 1, again, PBMCs, and we repeated this experiment. Number 1 is the green one, and you can see that, again, we have a nice recognition of this peptide, but no recognition of the other New York BR1 peptide or an irrelevant TRP2 peptide. All of these are DR3 peptides. And actually the other DR3 peptide was not recognized, which was a confirmation of the result here, because that was one of the patients which did not recognize this peptide. So all in all, we can say, yes, we found at least in 3 cases clear, and this is a little bit doubtful. Patients, CD4 T cells which recognize these class 2 peptides from New York BR1. So here I'm already at my summary. So we have identified New York BR1 specific HLA-restricted CD4 T cell epitopes. We could establish murine HLA-restricted CD4 T cell lines. We could confirm the endogenous processing of the newly identified epitopes in human cells, and we could detect New York BR1 specific CD4 T cells among the PBMCs of breast cancer patients. What can we do with all of this? So presently we are working with MHC class 2 tetramers, which have been provided by the NIH on all of these 4 epitopes. And of course here the aim would be to have a closer look at the CD4 specific T cells from patients in terms of different parameters, or to use these tetramers for immunomonitoring, which is also an important issue. And we have sequenced already the TCR of the mouse cell lines, but not of human cell lines. And of course, sequencing and cloning of a New York BR1 specific TCR would be a further step, either into the direction of TCR transfection of lymphocytes, or usage of the epitopes as helper peptides for vaccine development. And this is my final slide, so I would like to thank the people in my lab, especially Adriane Gardian, who did most of this work in her PhD thesis. And these are the collaboration partners from the gynecology and from the pathology, and also Dirk Jäger, who actually has invented that epitope, that antigen. Thank you very much for your attention.
I want to take a few questions for Dr. Eichmuller. Yes, Paul?
I was wondering with regards to TCR sequencing, whether you would plan to look at the regulatory T cell pool, because I always find that an interesting thing, is whether they're responding to the same epitope.
Right.
Yes,
Good
that's actually this is what we plan with the tetramers, to have a closer look at the human cells. We did some first experiments, especially in the mouse, but these were not really solid enough to present them here. So our first glance was it's probably not regulatory T cells, but we need a better proof with tetramers to really identify the individual cells. But that's a very important issue.
So why do you think that, I mean, all of the patients you're testing all have the DR. They all have the right HLA class 2, right? So why do you think that some are processing it appropriately and some are not?
Yeah, that's also a good question. So of course they differed in the subtype of DR, especially DR4. I don't know whether you saw the table. There was DR405 and 401, and I'm not 100% sure whether these are similar. But one case I pointed out was DR3, and I think in these cases they even had the same subtype. I can't answer.
Okay, so I'm going to end this session. I want to thank all of the speakers very much. I think it was a really exciting session. For those of you who are not cancer immunotherapists, I hope you feel that this is really, was the breakthrough of the year last year. And I hope, of course, we're all here to do the same thing, which is to cure cancer and to treat cancer. And we immunotherapists really feel that this is one of the therapies of the future. So I think our speakers today are going to be around in the coffee break if you've got some more questions. Thank you very much for your attendance.